afternoon. Today we're going to talk about acute infections. This is one of two lectures which talk about the two different patterns of infections that viruses present when they infect a host. So here's a graph that summarizes the two different kinds of general infections. And on this graph we have virus production on the y-axis with time on the x-axis. And the blue line is virus production. The red bars are periods of disease. So on the top is an example of an acute infection where you have a very defined period of virus reproduction and disease. And below are all different kinds of persistent infections. They fall into different categories depending on how they look, but they all have the characteristic that the virus remains with you for your lifetime. And your lifetime is shown here on the graph, death. That's the end. And you can see all of these three, which are three different kinds of persistent infections. They all last for the lifetime of the host in terms of virus and even disease. So for example, we have latent infections where you have periods of virus production followed by no virus and periods of disease or not, depending on the case. And we'll talk about some of these today. We have some virus infections which are persistent. They last your lifetime, but they're asymptomatic. Many of us are infected with a variety of viruses that don't cause any disease. And then we have some viruses where you have an initial acute, what looks like an acute infection, followed by a long period of clinical latency and then virus reproduction and death. And that's HIV-1, which we will talk about later. Today we're going to focus on acute infections. At the top, pattern at the top, an acute infection, rapid onset of viral reproduction, a short course of disease, but it could be serious, it might not be serious, could be inapparent, but could be serious as well. Production of large numbers of virus particles and immune clearance typically. So the virus is cleared at some point. You either die or the virus infection is cleared. So Ebola virus was considered for many years to be an acute infection. You either died or you cleared the infection. Now we think there may be some examples of persistence, and we will talk about that. Here's the time course of a typical acute infection, and it's presented in terms of host defenses that occur. So we here again have virus growth on the y-axis with time on the x-axis. And we have entry of the virus by all those mechanisms we talked about some time ago. We have innate defenses responding. The red line is the virus titer. So the innate defenses respond, but if they cannot prevent infection, they do prevent many infections, but not all. The infection becomes established, virus keeps reproducing, and the adaptive response is engaged. Virus reproduction peaks, and then the adaptive response will help to clear the infection by whether it's by antibodies or cellular responses or both. And eventually the virus is cleared, the host recovers, acute infection is over, and you have immune memory. Typical acute infection. Now, in acute infections, any infection, even persistent infections, we have a concept called the incubation period. So I want to define this for you. It is the initial period before symptoms of disease are obvious. Remember, symptoms are what you can feel. You can't feel virus reproducing in you unless it's damaging something and you feel the pain of that. But initially, when influenza first enters you, you don't know it's replicating. When you finally feel scratchiness, sore throat, whatever, those are symptoms. So before that period, that's the incubation period. So it's defined in terms of symptoms. Now during this incubation period, the genomes are replicating, viruses are replicating, the host is responding, the innate response is responding. You can't feel any of that initially. The virus may or may not be transmitted. So this can be an infectious period depending on the virus. Some viruses are transmitted during the incubation period, some are not. For example, Ebola virus is not transmitted until you show signs of infection, that other people can tell that you're infected. But other viruses are different. So that's the incubation period, all right? It's this period where viruses have come in, they're replicating, you're responding. If we measured virus replication in you, we could detect it, but you can't tell it's going on. So that's why we use symptoms as the delimiter. And then you go into the period where you have clear signs, you have clear symptoms of infection, and that is beyond the incubation period. Here are some incubation periods of viral infections, from very short to very long. So we have a few days for influenza viruses and rhinoviruses. And in general, but not always, when the incubation periods are short, that means the virus doesn't have to go far to cause the symptoms of the disease. So the respiratory infection, the virus enters the respiratory tract, it replicates there, and it's able to cause symptoms quite rapidly. And for long incubation periods, typically, again, but not always, it means that the virus has to travel somewhere else to cause the symptoms. So let's take measles. Okay, so 9 to 12 days. Remember, the symptom of measles that you can see yourself is the rash, right? And that takes a while because the virus has to go from the respiratory tract to the skin. You can see some of these are very long. Rabies, 30 to 100 days, depending on where you're bitten, before you start showing the neurological symptoms of infection. That's why we can immunize after you've been bitten against rabies, because the incubation period is so long. Okay, so that is incubation period. There's another term you may encounter. It's called the prodrome. It is the period of symptoms before those characteristic of the disease. So if you start to get fever and aches, that's the prodrome, because they could be anything. And let's say you have measles infection, you get fever, achiness, prodrome, and then you get rash, that's the symptom of the disease. That's the characteristic. So that's no longer the prodrome. Many acute infections are inapparent. This is a concept we've talked about before. You're infected, the virus is replicating, you have an immune response, but you don't feel anything. You have no symptoms. You would certainly have signs. Signs are what you could measure, so you could take out blood and see that the virus is replicating, but there are no symptoms. That's what an asymptomatic or inapparent acute infection means. Successful infections, no symptoms or disease. However, sufficient virus is produced to spread in the population. And an example is West Nile, which we talked about a few lectures ago, where many infections are inapparent and people are infected, the virus is replicating in them. Poliovirus is another example. Only 1 in 100 people become paralyzed. Most of the others don't show any symptoms or kind of mild, nonspecific symptoms. So you have no idea that they're infected and they're out there spreading virus. When I say these are successful infections, how would we know that someone is infected and show no symptoms? How can we conclude that they're infected? How can we conclude that they're inapparent infections at all? Does anyone know? Absolutely. You would find antibodies against the virus in the blood. So we do serological surveys of populations. Take serum from lots of people. If you want to do a study, you register the study, you get permission to do it. You go out and you take serum and you look for antibodies of the West Nile, and you can find a certain percentage of the population has been infected by the presence of antibodies in the serum. It's a good measure of infection. And you can also ask them, do you remember having any of the symptoms of West Nile? And they said, no, I never had any symptoms. I've been fine for the last 6 months. So they had an inapparent infection. That's how we know. We take serum and we conduct these surveys. Inapparent acute infection is the sign of a well-adapted pathogen. Pathogens that have been infecting people for many, many years, maybe thousands of years. So they came from an animal source. As you'll see later, every virus we have today came from some kind of animal, whether it be proto-humans or bats or something else. No viruses originated in us, and some of them originated and came to us many years ago. Measles, for example, is estimated to have jumped from cows to us about 12,000 years ago when we started raising cows to eat them and to drink their milk. We got them together in farms for the first time, and people were interacting with them. We got their viruses. It's all our fault in the end, you know. So well-adapted pathogens have been for us for a long time, and they tend to evolve to cause lots of inapparent infections, because when you think about it, think about this. There is no reason for a virus to kill you. It doesn't help it transmit. The sine qua non of a virus is to find a new host. There's no reason to kill you. Killing is accidental. It's either our immune response that's overexuberant. In the case of the Ebola virus, we have hugely overexuberant immune responses. Or the virus needs to replicate to certain titers in order to be transmitted, and in the process of replicating to those high titers, it kills you. But killing per se doesn't do any good for a virus. So over time, viruses tend to evolve to be less pathogenic, cause a lot of inapparent infections. A sign of a recently acquired virus, those are pretty virulent, they haven't had time to adapt. HIV was about 1900 spill over into people. And there's signs it's getting less and less virulent, okay? And I would predict in 100 years it's going to be way less virulent. But there's some viruses that never go that way for some reason. Smallpox never got less virulent, as far as we can tell. It always killed a lot of people. So I guess for some viruses to spread, they have to replicate to those extents. Okay, acute infections are public health problems because they infect millions of people every year. Infections like influenza virus, norovirus, rhinoviruses, and others. They're so fast. By the time you feel sick, you have already spread the infection, and it's likely to be almost over. So that's one of the issues with them in terms of public health control. They go so quickly. It's hard to treat them. Whereas, you know, chronic hepatitis B, hepatitis C, HIV, they go on for months, years, plenty of time to treat them, plenty of time to diagnose and treat them. So acute infections have this particular problem because they are fast. It's really hard to diagnose and treat them. And so as you'll see, we have few antivirals for acute infections. Most of them are for persistent virus infections. First question, which of the following do acute infections and incubation periods have in common? The virus is not replicating, no symptoms are visible, immune defenses are engaged, the immune system does not respond, or all of the above. So this is a tricky question, okay? Only 61% of you got the right answer, which is immune defenses are engaged. So the percentages are misleading because with only 30, 40 people, doesn't mean a lot. But first of all, what do acute infections and incubation periods have in common? The virus is not replicating. That's wrong. So good. None of you picked that. Great. No symptoms are visible. An acute infection at some point has symptoms, so that can't be right. I understand the way you were thinking about it. You think you're kind of confused by the incubation period where no symptoms are visible, but the actual acute infection, for the whole course of the acute infection, there certainly are. And the immune system does not respond. That's not true, because during the incubation period and the whole infection, the immune system is responding. So that's why it's C. So today we're going to go over 5 viruses that cause acute infections. I'm going to go through each one, how they infect you, the kinds of pathogenesis, immune response, what we can do about them, and so forth. They include influenza virus, poliovirus, measles virus, norovirus, and West Nile. I've picked them because they are different diseases, different patterns of infection, and they're transmitted in different ways. So, for example, influenza is by respiratory aerosols, person to person. Polio is fecal-oral contamination, person to person. Measles virus, person to person, respiratory. Norovirus, fecal-oral, person to person. And then West Nile is mosquito to people with a bird intermediate. So let's start with influenza virus, which we have talked about in terms of replication so far. And remember, it is an RNA virus with an envelope-negative stranded RNA with 8 genome segments. There are 3 types of influenza virus. They're called A, B, and C. A and B will cause a similar disease that's known as influenza. C is mostly inapparent or mild upper respiratory tract illness. We only vaccinate against influenza A and B. And only influenza A causes pandemics, which is a global epidemic where a new virus is introduced, it spreads globally, and many, many people get infected. B does not cause those pandemics. And a highlight of influenza is that it undergoes extensive antigenic variation from year to year and every 20 or 30 years as well. It's transmitted by aerosols produced by coughing, sneezing, and talking. I would say mainly by talking. We're giving out these aerosols, some of which are very small and can travel long distances, and others are larger and don't get very far. You can also contact individuals. If you get some mucus from someone on your hands and put it in your nose or your eye, you will transmit the infection. And you could also get it from a contaminated surface, depending on how long the virus has been. It's not going to last forever on the surface, but it will last a few hours. And some surfaces have a lot of traffic, like doorknobs. People touch them with hands that they've had in their face, and so they can transmit viruses that way. But certainly respiratory aerosols are a big part of it. The virus is inhaled in those aerosols. It goes into the upper tract, infects the respiratory epithelium, and then the virus spreads laterally through the epithelium. It does not cross the basement membrane. It stays in the epithelium. This is a restricted infection for the most part. There are a few exceptions. And it eventually will start moving down the tract. In some people, it's restricted to the upper tract, but not always. For most, it moves into the trachea, and you can feel it in your chest. You have a very strong sensation of discomfort focused on the trachea, and that's where the virus is replicating. And if you're unfortunate, it will proceed down to the lower parts of the lung and can even cause a pneumonia, which is very serious. So it's a respiratory disease focused on the trachea, but can spread throughout the tract. If you remember from when we talked about virus entry, the receptor for influenza viruses is sialic acid, a sugar which is a terminal sugar on viral glycoproteins. Sialic acid can be linked to the second sugar in a glycoprotein by multiple chemical moieties, and two of them are shown here. Alpha-2,6 or alpha-2,3. So the alpha-2,6 linkage is predominantly recognized by human influenza viruses and the alpha-2,3 by avian. There are lots and lots of avian influenza viruses out there circulating in wild birds, mainly water birds, but also in others, and also in farmed birds, chickens and geese and ducks and so forth. And they're a huge reservoir, and they transmit the virus quite readily, and humans can get infected with them. But those avian viruses prefer an alpha-2,3 sialic acid linkage. Now, we have some alpha-2,3 sialic acids in our respiratory tract, so occasionally people do get infected with avian influenza viruses. So here's a picture of the tract with the alpha linkages indicated. So you can see in our upper tract we have all alpha-2,6, nasal mucosa, pharynx, larynx, etc. And so that's where the human viruses initially replicate. And then down in the trachea there's a mix of alpha-2,6 and alpha-2,3, the same with the bronchus and the bronchioles. Human viruses can replicate throughout the tract. Now, avian viruses are not likely to infect the upper tract. However, there are alpha-2,3 sialic acids in the eye. You can see here the cornea, the conjunctiva. And so it's been shown that avian influenza viruses can infect the eye. They will then drain through the lacrimal duct, nasolacrimal duct, into the upper tract, and then they can eventually spread to the lower parts. And so this is how humans can get infected with avian influenza virus, either in the eye, or if you inhale very deeply and take a deep breath of an avian influenza virus and get it into your lower parts of your lung. Now, you may say, how likely is this? Well, in many countries where people raise, say, chickens, they sleep with the chickens. They're in the same house. And often kids get avian influenza viruses by sleeping next to the chickens and inhaling that exhaled virus all night. So it can happen. Here are is the progression of uncomplicated influenza, short incubation period. You can usually pinpoint when you've got the first Symptoms of influenza, you get headache, chills, and dry cough, which are prodromal. Can't really tell what kind of infection that is. Initially, you get a high fever, myalgias, malaise, anorexia. Fever peaks within a day and then declines. But as the fever declines, you then get an intensification of the respiratory symptoms. Your cough goes from dry to productive. You notice later, after you're recovering from flu, you're coughing up big gobs of mucus and you're trying to clear the virus. And this really wreaks havoc on your respiratory tract. The combination of virus replication and CTL, CD8 positive T cells, lysing your respiratory epithelium, trying to clear the infection, makes you really feel badly. Many people have trouble breathing. Even if you've already have a compromised breathing, it gets even worse. And this can last for a long time because it takes time for your respiratory epithelium to heal. Nowadays, we can diagnose influenza in a couple of ways. It's called influenza-like illness. So the CDC says all you need is fever of at least 100 degrees and cough or sore throat. And if you are in the influenza season, which is the fall through March, April, or May or so, no other known cause, they say it's influenza. There are rapid lab tests, but their accuracy is not very good. Better accuracy is to do PCR or viral culture or serology, but of course those take time, and this is, if you want to take an antiviral for flu, you need to know you have flu within a day or two, otherwise an antiviral is not going to help you. So that's why we use these rapid lab tests, but most of the time you go to a clinic in the middle of the winter, you have fever, cough, or sore throat, they'll assume you have flu and give you an antiviral without even testing for it. Here are a couple of experimental flu infections to show you the course of infection, symptoms versus virus. So on the left, we have inoculated volunteers with influenza. We can do this with influenza because some of the strains are quite apathogenic, and we typically use people who have been vaccinated. So we inoculate here on day 0. We're measuring virus titers and symptom score. So you devise a numerical score for cough, sore throat, malaise, etc. And here we see viral shedding is in the black. So the first virus is, you can see, shed on day 1, and we have mild symptoms on day 1, and then viral shedding peaks between day 2 and 3. Symptoms are a little bit delayed behind it. So this, you see, is a rapidly resolving infection in the sense that the peak is very early and then the virus is declining. So unless you get the antiviral on day 1 or 2, it's not going to really change the outcome unless you are very old or immunocompromised, and the infection will continue longer. On the right is another experiment in humans where they have been given virus on day 0. We are measuring virus again. You can see a peak, same time roughly here, 3 days, rapid decline. Look at the interferon titers were measured, in the nasopharyngeal wash. And then we're looking at serum neutralizing antibodies. So the virus is going down way before the antibodies are going up. So the antibodies are not helping you recover. It's the cytotoxic T lymphocytes that come up quicker and resolve this infection. Influenza is a seasonal disease. Here's a graph of incidence. This is all from the CDC. They keep very close look on influenza every year, throughout the year. They get samples from all sorts of regional hospitals, and they ask what kind of virus is in it. Keep really careful records because this is a serious illness. And you can see here 2004, 5, 6, 7 peaks of illness at specific winter months. These are the weeks of the year. That's week 50, week 8, 18, and so forth. So these are the late fall and winter months where it's peaking every year. So very seasonal disease. And here are some more interesting charts from the CDC website. This is from last season, the influenza positive tests. You can see it peaking here in week 4 or so, the beginning of January. So you can calculate when this is. So starts very low, peaks, and then goes down. The colors are the different types. A is in red. There are a couple of different subtypes of A, H3N2 and H1N1. And you can see the B is in green. So in 2017-18, the H3N2 predominated. But look at this year. This is this year's season, which of course is not over yet. The H1N1 subtypes are predominating, but the same pattern. On the upper right is pneumonia and influenza mortality from year to year, 2015, 16, 17, 18. This is where we are now. And so the black lines are the calculated maximum minimum of mortality from respiratory illness. And then the red is actual data from flu that year. So you can see in some years there is excess mortality, which means there's a particularly virulent virus circulating. So back in 2014, last year there was a peak of excess mortality. This year it is not such a virulent strain. Now on the bottom is a really cool graph of the US where they go through every month and they tell you the activity in each state. And you can go from minimal, the green colors, up to red. And so I made a little movie of this to show it to you. Let's see if it works. Yeah, so now we're going to start way back in the fall. And you can see each state starting to get redder and redder. Other states not so, more and more, until that's the peak of the epidemic, and then it's going to go down. Some states are returning to green now. So you can see throughout the US, obviously. And then finally, another way of looking at the data, this is percentage of hospital visits for influenza-like illness reported by outpatient units throughout the US. So they have a network of facilities who report specifically to the CDC. They tell them what's happening in terms of visits for flu. They send them samples and so forth. So you can see these are each colored line is a different season, and percent visits for influenza-like illness. You can see back in 2009, the gray line, there was a huge peak. That was a pandemic year where we had a new strain, so lots of visits for it. In intervening years, you know, fewer numbers. Let's see where we are now, 2017. Last year was pretty serious in terms of admissions, but this here we are, 2018-19, sorry. This one is lower, as you can see, which was reflected on the previous excess mortality graph. We get about 30 to 50 million cases a year in the US. It's a lot of infections. Not all of them are serious, of course. And between 3,000 and 49,000 deaths. That's the range for the past 31 years. Some years, a few thousand deaths. Some years, 49,000 deaths. And people always tell me, I don't want to get the flu vaccine. I never get flu. It's really silly not to get it. There's no negative. It doesn't work great, I know that. The efficacy is not what it should be. But what if you died 1 year from flu? You would regret it. Well, you don't regret it, right? Maybe your family would not be happy. So I'm always immunized. My family is immunized because you don't want to be one of these statistics. It can kill a healthy student like you, a healthy young student like you. So you should get the vaccine every year. You can get complications of flu. You can get viral pneumonia where the virus goes down into your lower tract and causes serious infection. You can get secondary bacterial pneumonia afterwards, muscle pain. You can get your heart involved. Some people whose breathing is so impaired that it puts a stress on their heart and they can have heart trouble. And it can also cause what's known as Reye syndrome, which is encephalopathy and liver damage. You can take drugs for influenza. You can go to the drugstore and waste your money on non-pharmaceuticals, or you can get antiviral drugs, but if you, and here are 3 of them, if you don't take them on day 1 or 2 after that first symptom, scratchy throat, whatever it is, you're not going to get any benefit from them. So if you feel in the winter scratchiness, go right away and get a script if you want to take these. They do help, actually. They reduce symptoms. And, of course, there are vaccines to prevent infection. All right, this is not as tricky. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. Incubation period 1 to 5 days. Fever peaks within 24 hours. Coughing and weakness can last for 2 weeks, all of the above. 95%, not bad. It's all the above, of course. Everything is correct. Okay, our next acute infection is poliovirus and poliomyelitis. We've talked about polio as a model virus, small, spherical, non-enveloped icosahedral particle, with an RNA genome plus stranded polarity. It's the virus I spent most of my career working on. The pathogenesis, you ingest fecally contaminated material. And it still happens to this day because in general, humanity has really bad hand hygiene. We go to the bathroom and we don't wash our hands, and then someone else gets our fecal material. And kids are particularly good at this because they don't wash their hands at all. But it's not just poliovirus that can be transmitted this way, other viruses as well. You ingest the virus, it passes through your stomach, so it's obviously resistant to low pH, and then alkaline pH in your intestine. It ends up in the intestine, small and large intestine, replicates in the mucosal epithelium. Virus is shed into the lumen, and that's how you excrete huge quantities of virus in the feces, and that's how you spread it to another person. In 1% of infections, 1 out of 100, the virus gets into the central nervous system. So the virus routinely goes past the basement membrane, gets into the circulatory system, establishes a viremia. And, in 99 out of 100 patients or infected people, you either get a nonspecific febrile illness or nothing. And only in 1% of people does the virus get into the CNS and destroy neurons and cause limb paralysis, hands or legs. And we think the virus goes from the blood to the muscle. In the muscle, there are nerve endings. The virus is taken up, travels to the CNS through the nerves and reaches the motor neurons in the spinal cord. These, of course, are wired to primary neurons in the brain, so the virus can replicate in either in destroying them. You destroy enough neurons, you get paralysis of the leg. But that happens only in 1%. It's an accident. The virus need not go into the CNS because it doesn't get out once it gets in. So it doesn't transmit. There's no selection for the virus to get into the CNS. It is an accident. Yet, during the big epidemics of polio in the US, here in New York City, we used to quarantine paralyzed kids. Police used to go from door to door in New York City, knock on doors and look for a paralyzed kid and drag them to the hospital. Absolutely useless. Well, we've realized that at some point because there's one kid there shedding polio, but it's not very mobile, so how much contact with others is he or she going to have? The 99 other people are infected without any symptoms, are walking around spreading the infection. So you can never succeed by quarantining the 1%. Of course, in hindsight, everything is 20/20, right? But it was really, for most virus infections, in fact, quarantining doesn't work. Not for all of them, but for that one, it didn't make a difference. Here's a section of a spinal cord. This is from a transgenic mouse, the model that I told you about last time that we made for studying polio. And this is from a mouse that was paralyzed. And you can see these dark staining cells. These are immune cells that are coming into the cord trying to eliminate infection. And the infected cells are in green. This is virus RNA, which is what we've stained here. These are neurons. You can see big pyramidal looking cells. They're all neurons. And if you get enough of those destroyed by probably lytic viral infection, we don't really know, then that muscle that's innervated by these neurons will be paralyzed. Here's a single neuron, so you can see full of viral RNA, cell body, axons, and dendrites. So again, this is only 1% of infections. It's an accident. But of course, it's an accident that drove the development of 2 vaccines, and now we're about to eradicate it so kids don't get paralyzed, which is good. Here is an old graph of the progress of polio infection, which I really love. It's from a 1950s textbook of medicine. Those of you go to medical school, try and find polio in your textbook of medicine. I don't think you'll find it there anymore. And this is hand-drawn. In the old days, we didn't have computers to draw figures. We had to draw figures by hand. And so here's a clinical course. Exposure to the virus happens here at day 0. We're looking at time and different parameters on the y-axis. And so here we're looking at temperature, clinical temperature here in Fahrenheit. You have a spike of temperature at about day 7. So the virus has replicated in the gut, it's established a viremia, and now you're making cytokines that induce a fever. Day 7, headache, sore throat, nausea. 99% of people after that get well. the viremia goes away, the temperature goes down, and they're fine. So at the same time, if you look at virus and throat secretions, you can see it's peaking around the same time. And so is virus in the feces. Look at virus in the feces goes for a long time, up to a month here, and can be longer. Virus in the blood peaks there, and your antibody then comes up later. All right, so in 1% of people, the infection continues. There's another bigger peak of temperature days later. About 10 days later, you get headache, nausea, stiffness, pain, and paralysis. In these people, the virus has gone into the CNS, higher temperature spike, and you look down here, you can see virus in the CNS, which didn't happen earlier. It only happened later after most people recovered. You see the paralysis right there. So that's the course of infection. And again, beyond this initial phase of nonspecific illness or no illness, only 1 out of 100 people move on to paralysis. But everybody secretes virus in the feces, so they're all eligible to spread it. We're the only known reservoir of this virus. That's why we can eradicate it. Once we eradicate it from humans, there's no other reservoirs to spread it back, spread by fecal-oral transmission. This is a disease of warm months in temperate climates like ours. Sometimes there is a long-term complication called post-polio syndrome, which happens about 30 to 40 years after the initial paralysis. So in many individuals, you can recover limb function by therapy, and these individuals walk and do everything normally. And then 30 to 40 years later, about 25 to 40% will develop paralysis again. It is not an infectious process. It is because as you age, you lose neurons naturally. And when you've had polio, you don't have a backup anymore. You destroy a lot of neurons with polio. So as you age now, you lose more than you're able to sustain movement with. So we still have people developing post-polio because we're still in the era where there was polio these many years ago. In the US, we used to have 20 to 30 to 40,000 cases of paralytic polio every year. We developed 2 vaccines, which we'll talk about later in some detail. An activated vaccine introduced in 1955. In fact, on April 12th or April 15th, will be the anniversary of its licensure in 1955, the Salk vaccine. That brought the number of cases down to about 2,000 a year. In 1962, we introduced the oral Sabin vaccine. The last case of polio in the US, 1979. We haven't had a case of polio since then. And as we'll see later, globally, there are less than 200 cases a year of polio. And so it is on the verge of being eradicated. And it probably will be within 10 years or so. So here we have measles virus. So we go back to an envelope virus, negative stranded RNA, single piece of RNA in the particle. This is also a virus that is transmitted by respiratory aerosols. So we go back to respiratory transmission. And this virus is one of the most contagious human viruses. We're going to talk about R0 a bit later, but this is a parameter that tells you, on average, one infected person, how many people can that person infect? And so for measles, it's 15, which is very, very high among the viruses that we know of. In fact, if you've seen the movie Contagion, there's a scene where they're talking about R0. Pathogenesis, 1 serotype only. So for polio, there were 3 serotypes, so we've had to make a vaccine with 3 different viruses in it. For measles, there's just 1. And by the way, we've been using this vaccine for over 50 years, and it still protects against measles virus. So there's no antigenic variation. Infection gives you lifelong protection. This is transmitted by respiratory aerosols, very much like influenza virus. You are actually contagious before the rash appears. So you are exhaling virus before you have obvious symptoms of measles. So this presents a problem in school situations, right, where kids can be sitting in class exhaling measles virus. Now, we don't have that much anymore because kids are vaccinated for the most part, but in the days before vaccination, it was how transmission occurred. And here, in contrast to polio, nearly all infected individuals show signs of disease, i.e. the rash. How this infection develops: you inhale respiratory aerosols containing the virus. Virus replicates on mucosal surfaces, goes through the lymph, establishes a viremia, and that brings virus to the skin, causing the rash. And in some cases, it can get into the brain and cause encephalitis. And it's one in a few thousand cases you can get measles encephalitis. So it's not an insignificant complication. How the virus gets in and out is very interesting. So here's the detail on the right of that. The virus comes into the respiratory epithelium, doesn't actually enter the epithelial cells. There are no receptors on the apical side of the epithelium for this virus. Rather, the virus enters immune cells, lymphocytes, macrophages that happen to be present in the epithelium. And that's shown by this orange cell here. So the virus infects it, and those cells go below the basement membrane and bring the virus elsewhere. This virus likes to replicate in lymphoid tissues, so it has a good time in the lymph nodes and in other parts of the body. You get a viremia, and then that brings the virus back to the epithelial cell surface. Remember, there are blood vessels below the epithelial sheet in our mucosal surfaces. So there's measles virus in those blood vessels. It gets out of the blood vessels, and it moves to the basolateral surface of the epithelial cell, where there happens to be a receptor for the virus. So these respiratory epithelial cells have no receptors on the apical surface, but they do on the bottom. Virus binds to them, replicates in the cells, and gets out. It's a brilliant strategy for spreading, right? Because without that receptor on the basolateral surface, it wouldn't be able to get out and spread to another person. Again, no receptors on the apical surface. The virus gets in via mobile immune cells. And of course, the respiratory aerosol then transmits it to other people. Here are some of the symptoms of uncomplicated measles: high fever, runny nose and cough, conjunctivitis. Koplik spots are little white spots in the mouth of someone who has measles. These are actually syncytia. If you took a little scraping of these and looked at it under the microscope, you would see giant fused cells. And in the days of measles, you could actually say, oh, this kid has Koplik spots, he or she has measles, rash from face to the extremities. There are complications of measles that are not insignificant. Post-infectious encephalitis, 1 in 1,000. 1 in 1,000 infections. Kids, the virus can go in their brain, cause problems, and even if they recover, they can have long-term issues with that. Bronchitis, pneumonia, ear infections. The fatality is 1 or 2 in 1,000, but as I said last time, in countries with poor nutrition can be up to 30% mortality after measles infection. A long-term consequence, which we'll talk about next time, is a disease called subacute sclerosing panencephalitis. These are kids who recover and then many years later develop a chronic degenerative neurological disease. And of course, this virus immunosuppresses. We talked about how it does that last time. It replicates in various immune cells, including APCs, and you get secondary infections, and that is the main cause of death in undernourished children, secondary infections. So in the heyday before vaccination, we used to get 3 to 4 million cases a year of measles, 400 to 500 deaths, a lot of hospitalizations, a lot of chronic disability from encephalitis. This is why we developed a vaccine. Lots of people worked hard to do this. It's a very safe vaccine. We stopped endemic transmission in the US by the use of this vaccine, which was introduced in the 60s. It's introduced as part of the measles, mumps, rubella vaccine, 3 viruses altogether. However, in 1998, of course, the Wakefield report, which suggested that the vaccine was causing autism, was completely wrong and had been withdrawn. That led to decreased immunization, outbreaks in Europe, and we still suffer the consequences of Wakefield's nonsense and poor science because people believe him or they don't know any better. I don't understand, but there are outbreaks all the time. There's currently one in Rockland County you may have heard of. Quite a few, over 200 or 300 cases of measles and individuals who don't immunize their children. And there's no reason for this. They have actually imposed a ban on kids under 18 years of age being in public unless they're immunized because it's so serious in Rockland County. This is a safe vaccine that prevents measles infection. There's zero reason not to use it. There is no reason to debate its use. It should be used, and if you are ever physicians one day, you should make sure your patients are immunized. Don't give a medical exemption unless it's warranted. And only 1% of the US population warrants a medical vaccine exemption. So here are the cases of measles in the US, vaccine licensed in the early 60s, and you can see it really dropped the number of cases, but there are always blips because of failure to immunize in groups of people in the US, and then virus is imported from Europe and causes outbreaks. And we've also decreased the measles deaths per year as a consequence. Here are some recent numbers on measles from the CDC. So these are cases of measles by year 2010 to 2019. And you can see not a lot, 60, 200, 187. Big outbreak in 2014. I think there was one in Disneyland in California, where a group of communities whose parents decided they didn't need to immunize their kids, they all went to Disneyland and they started a big outbreak. And now in 2019, we have 314 so far, and these on the bottom are 6 outbreaks currently ongoing in the US in different states. These are linked to travelers who brought the measles virus back from other countries where large outbreaks are occurring. And if you go to travel, when you go to the airport, you'll see signs which say, have you had your measles vaccine, because there are outbreaks in Europe. I can't tell you how inexcusable this is. Now, in many parts of the world, as I'll show you, people don't get the vaccine. But in the Ukraine and in Israel, they should be getting the vaccine. And certainly in this country, everyone should be getting it. And for some reason, people still argue that they want a decision on what to put in their bodies. To which I say, you have no decision to make someone else ill. So you have to be vaccinated or you have to stay home. When it comes to infectious diseases, it just doesn't work that way. You need a license to drive your car, right? People don't complain about that. You need a vaccine, they complain about it. I don't get it. You explain it to me. This is the global situation. Many countries do not have access to vaccine. And so here are the numbers. The darker the color, the more cases. And the US, we used to be white, but now we are orange, because we have, as you saw, some cases. Lots of countries in Africa and Asia where people don't get the vaccine, but other countries where the vaccine is given and many people choose not to get it. So Ukraine currently is in the top 10, India, Madagascar. You can see the numbers of cases and the rate of infection. So these are pretty recent data through March 2019. So in 2014 alone, we prevented 115,000 deaths from measles by vaccinating. In this 14-year period, we prevented 16 million deaths. And I like to say this is one of the best buys in public health. Yet it's the target of people who don't want to be vaccinated. I'm completely baffled. All right, the next question. Which of the following is a good reason to get measles vaccine? There is a 1 in 1,000 chance of acute post-infection encephalitis, and 1 to 2 in 1,000 chance of death. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections, all of the above. All right, so most of you got all of the above, which is the right answer. All of these are right, and they're all things that I've touched on just now. Let's move on to another virus. Here's a lovely statistic I learned from a gastroenterologist at Stanford. In a 24-hour period, 200 million people in the world have gastroenteritis, and the amount of water they pass equals the water passing over Victoria Falls in 1 minute, which is 65 million liters per minute. That's how much diarrheal volume is being passed by these people with gastroenteritis, which is an inflammation of the intestinal tract, mostly caused by viruses, although some bacteria can do it as well. And mostly the gastroenteritis is caused by a norovirus, which is a member of the virus family Calliciviridae. These are plus-strand RNA viruses with naked icosahedral particles, very, very much like polio in their overall structure, but in a different family. They cause 50% of all foodborne outbreaks of gastroenteritis. That amounts to 23 million cases a year of gastroenteritis caused by food. So here is a breakdown of, it's an old slide, known causes of foodborne illness in the US. You can see norovirus 50%, bacteria 40%, and then some other causes, parasites, chemicals, etc. So norovirus is the top cause. And before we knew about norovirus, we thought it was all bacterial gastroenteritis. But then we saw virus particles in the diarrhea of people with this, and that led to their discovery. Norovirus is what I call a two-bucket disease. Right? You need two buckets, one for the vomit and one for the diarrhea. 2 buckets. It's a real term. Fecal oral spread, it goes through your stomach, so it maintains infectivity, infects your intestine and causes blunting of the villi in the proximal jejunum. So here on the bottom are 2 sections of villi from this area. This is an uninfected on the left. You see nice villi, and they become blunted when they are infected with norovirus, and that probably contributes to the gastroenteritis and diarrhea. We don't actually know what causes the vomiting and diarrhea. You remember I told you previously that for rotaviruses, there's a viral toxin that can reproduce the diarrhea on its own, but we don't know the cause for noroviruses. What's really interesting is that it turns out that for many years we couldn't grow these human noroviruses in culture, in cells in culture in the laboratory. Nobody could grow them. You could take diarrhea from a person with norovirus gastroenteritis, filter it to sterilize it and put it on the cell culture. It never grew. The virus never grew. Well, guess what? Turns out you need the bacteria for the virus to grow. And once someone made a mistake and forgot to filter the stool specimen, put it on cells, and the virus grew. So here's an experiment that shows you that bacteria are needed to promote infection. And this virus happens to infect B cells that are in the gut. Here we have viral genome per well with days post-infection. So we're infecting cells using filtered virus. In the gray, you see very little replication because we've removed the bacteria. And here, unfiltered virus has bacteria in it. So you need bacteria in your gut in order for norovirus to infect. Sort of like polio I showed you last time, requires a gut microbiome to infect. So it's kind of an emergent theme that these gut viruses have evolved with the microbiome as well. And what we think happens is that here's your gut mucosa, epithelial cells, Peyer's patches, collections of lymphoid cells, so lots of B cells in there. Here's norovirus, the red virus particles in the lumen. They bind to a carbohydrate on the bacterial surface. Bacteria will get transcytosed through the epithelium, probably in M cells, those green cells there. They end up in the subepithelial space, and then they can infect B cells. And B cells are essential for the reproduction. Virus probably reproduces elsewhere as well, but that's why the bacteria are essential and why no one could grow the virus, because they were filtering the bacteria out. So how contagious is it? 18 viral particles can infect you. People have done this study. It's very low amount of particles. And what's on the head of a pin apparently can infect more than 1,000 people. And we know this because you can infect people and do studies. And these are often done at medical schools. They infect volunteers, they recruit volunteers, and they infect them on Friday. They give them a stool filtrate from someone else who's been sick, and they put some flavoring in it so it tastes better. And they infect them on Friday, they get sick on Saturday, and by Monday they're well and they can go back to class and they get $300 for this. But now, of course, that we have cell culture grown virus, we don't need to feed people stool anymore. I suppose $300 is worth it, right? So it's fecal oral transmitted, but also vomiting is known to transmit it, contact with contaminated material. Foods can be contaminated at the source. So oysters, for example, filter feeders can pick up the virus in the waterbeds. Raspberries, if you wash them with contaminated water. Food handlers are often asymptomatically infected, and because they don't have good hand hygiene, they can contaminate the food, and then you go to eat it, and you get sick. You know, in restrooms in restaurants, they have a sign on the wall, employees must wash their hands, right, before returning. Do you know why that was put there? Because of norovirus. I remember a time when those weren't in restaurant bathrooms. You probably don't, you're too young. It was a time when they weren't there. It was before we knew about norovirus. Pretty short incubation period. You know when you have a norovirus, you start vomiting. And you often get it from kids. When my kids were in school, they brought it home to me. And I said, shit, I have norovirus. Diarrhea, stomach pain. Pretty short duration of illness, although if you're immunocompromised or older, it's going to last longer. And 30% asymptomatic infection. So the food preparers, you know, they go to work, they don't feel sick, but they're shedding virus and they contaminate the food. And you eat something at the buffet and you get norovirus illness. So virus shedding can persist for a long time. So those food handlers can infect a lot of people. And it seems to be likely that you can get reinfected, although even as you're older. So I could get a norovirus gastroenteritis today. For some reason, immunity doesn't persist, and it may be that the secretory IgA in your mucosa, which is prime for preventing infection, that wanes too quickly to be preventive. This seems to be a human-specific virus, but there is some evidence that marine mammals are infected with similar viruses. They may be able to infect us. Affects everyone year-round, peaks in cold weather, and the outbreaks often occur in all these semi-closed environments, which are increasing in numbers in the last 50 years, right? We never had daycare centers when I was a kid. There was no such thing. And now we have lots of them. So people get together in these and they transmit infections. And here are what you can do about it. Mostly we just want to prevent dehydration, make sure you take lots of fluids. There are vaccines in development. We'll see how good they are. And there are deaths globally every year, mainly in children who get dehydrated, don't have proper fluid supply, and that's why they die. So here are the settings where you see norovirus infections. The vast majority is in restaurants, especially catered meals. Again, someone is contaminating the food. Retirement centers, nursing homes, and hospitals, etc., daycare centers, cruise ships, only 10%, although you hear about a cruise ship when it comes back because of norovirus. And the mode of transmission, mostly person to person, and foodborne is only about 10%. It can be transmitted by vomit. Here's an article about an airplane where a sick passenger threw up, right? The crew had to clean up, and they went on different flights, and they infected people on those subsequent flights. And so the aerosol produced by vomiting can spread this. So the next time you hear someone vomiting on an airplane, run really far away, because you could inhale it and swallow the vomitus. And as I said, you hear a lot about cruise ships coming back because of norovirus outbreaks. And here are just some of the lists of different cruise ships, sailing dates, and the causative agent. Mostly norovirus, but also sometimes gastroenteritis by an unknown agent. And again, these ships go out with thousands of people on them, 4, 5, 6, 7,000 people. And then there's an outbreak and they have to come back, get everybody off, sterilize the whole interior. It's really costly, and I can't think of a worse way to spend a vacation. Now, why are they associated with cruise ships? It's only 10% of all the outbreaks, but we track illness on cruise ships, so we find them and report them. Close living quarters, everyone shoved in together. Food handlers secreting virus with no symptoms, new passenger arrivals, and so forth. So it's a closed environment, basically, perfect setup for food-transmitted infections. So how do you protect yourself? Wash your hands, rinse your food, cook your shellfish, because you can get it from them. I stopped eating raw oysters years ago, even though they're great. I don't want to take a chance. Although if you really like them and you don't mind being sick for 3 days, why not? And if you're sick, don't help others. And don't eat the mints in the restaurant. Don't touch them. They have done studies of these mints. they're completely contaminated with E. coli because people go to the bathroom, they don't wash their hands, and they take a mint on the way out, and they get shit in the mint, all right? So don't eat them. Even if they're wrapped in plastic, you're still going to contaminate your hands. I got an email yesterday from a student who took this course a couple of years ago, and she said, I still think about your lecture when I go for a mint in a restaurant. So don't eat them. This is a prime way to get contamination. Okay, last virus, West Nile virus. This is a virus we've mentioned a bit. It's a flavivirus. This one isolated first in 1937. It's called West Nile virus because it was isolated from the West Nile district of Uganda. Everyone thinks it has something to do with Egypt. So here's a cartoon from the New Yorker, right? We're pretty sure it's West Nile virus because this guy looks like an Egyptian, right? It's not. It has nothing to do with Egypt. We didn't have it in the Western Hemisphere until 1999, where it came in to New York City that summer. And the virus was sequenced, and it's identical to a virus from an Israeli goose. And so someone brought it from Israel, not the goose, but either a person or a mosquito got on the plane. Either one, that's how it got here. And it affects many different kinds of birds, many different kinds of mosquitoes, and other vertebrates besides humans as well. So this is the transmission cycle, primary cycle, mosquito, bird, mosquito, bird. So that's how it exists in nature. Incidental hosts, mosquitoes go and bite a human or a horse. They get infected, but they're not part of the chain of transmission typically. So often and outbreaks of West Nile are often coincident with drought. So when there's not a lot of rainfall, the birds go away, and who's left for the mosquitoes to bite but humans? And so that's how they transmit it to us. So you get bitten by a mosquito, short incubation period, you get a flu-like illness called West Nile fever. 80% of people have no symptoms, but of those 20 to 30%, they can develop severe neuroinvasive disease, which involves headache, eye issues, muscle weakness, cognitive impairment. A lot of these individuals have long-term cognitive issues, polio-like flaccid paralysis. So this is a serious infection, so we're working on a West Nile vaccine because if you're outside in the summer, in the warm months, you're at risk for being bitten by a West Nile infected mosquito. This is the pathogenesis of infection. You are bitten by a mosquito. The virus ends up in your capillary. It is carried by dendritic cells to the local lymph nodes. The virus likes to replicate in the lymph nodes. It establishes a viremia. It will then infect lymphoid cells and other tissues like the spleen. And remember, we talked about how the virus is sensed by toll-like receptor 3. That leads to the production of TNF-alpha, a cytokine that has the effect of loosening up the blood-brain junction. So the virus gets into the CNS, where it can replicate and cause damage to neurons, and that's why a certain number of individuals, 1 in 50, develop neuroinvasive disease and have long-term sequelae because the virus is damaging neurons, very much like poliovirus does. So this is not a goal of the virus to infect neurons because the CNS is a dead end. It will never get out of there. It happens to get in because it's an accident that TLR3 loosens up the blood-brain barrier. And all these cells here, just to indicate what are the cell types that are important for protecting the CNS against infection. Here are some statistics for you to look at. This is numbers from the US from 99 to 2013. So 99, the first year it came to the US, and this is broken down into total cases and neuroinvasive. So you can see in some years we have many thousands of cases, and lots of neuroinvasive cases, and other years fewer. It really depends on the ecology these particular years. As I said, some years it happens to be drier. That seems to increase the transmission. Here is the number of cases per 100,000 population with year up to 2017. So again, you see there are high years and low years. The week of onset is typically in the summer. Here's the number of cases versus the month of the year. And you can see from June through October, really, that's the peak time that we see West Nile, because that's when the mosquitoes are about. And so probably the best way you can prevent infection right now is to try and avoid encountering mosquitoes. So this is a neuroinvasive disease by county from 99 to 2017. The darker counties have the more neuroinvasive disease. And you can see where most of the West Nile cases occur, in the middle part of the US, where there are lots of mosquitoes and the climate tends to be drier. Here's the disease, neuroinvasive disease by age group, showing you that the older you get, the more likely it is that you are going to have neuroinvasive sequelae after West Nile infection. So neuroinvasive disease very low in the younger individuals, but 50 and 60 years and up, it goes up significantly. And that's, again, related to immune responses in older individuals and the inability to keep virus out of the CNS. So the main transmitters of West Nile in the US are the culicine mosquitoes, the Culex species, and these bite from evening to morning. So some mosquitoes are out during the day. Those are problematic because that's typically when you like to be outside, but this is a night biter. So if you want to go sit on your porch at night, you should take precautions. Now, here in the US, we don't sit on porches much anymore. If it's hot out, we sit inside with the air conditioning on, we have screens on our windows. But in many countries, people sit on the porches and they get bitten, and that's where there is more West Nile transmission. We are developing vaccines against this virus. This is 30% fatal in horses. And so people love their horses, especially race horses that bring them a lot of income, right? So a number of vaccines have been developed for West Nile and horses. You can go do a Google search, you can see all the different kinds. There are inactivated vaccines, there are even DNA-based West Nile vaccines, because it's a lot easier to get a vaccine licensed for a non-human animal than for humans. So they're being worked on in humans, and at some point they'll be available. They're not going to be required for everyone. It'll be required for people who are at risk. So if you're working outside all the time, in the evening particularly, you'll want to get this kind of vaccine if you do a lot of hiking and camping and so forth, etc. Let me end with this thought about viruses in the central nervous system. I've talked about poliovirus, measles virus, West Nile virus, all getting into the CNS in a fraction of infections. And I've already mentioned how they're transmitted. They're very effectively transmitted by shedding, here in the case of poliovirus, the gut, or measles virus, the respiratory tract, West Nile, by mosquitoes. They don't need to get into the CNS. In fact, I would say that CNS invasion of any virus is a dead end in humans because it cannot be transmitted to someone else. Now, in animals, it's a different story because animals eat each other and they will eat the brain and the spinal cord, which is infected, so that can transmit the infection. But in humans, the CNS invasion is a dead end. It may be a remnant of the old days when we used to eat each other, which we did at one time. Apparently we ate Neanderthals and so forth. And before that, when we were pre-humans, we ate each other. So the viruses that have stayed with us that whole time may have this remnant of getting into the CNS, which may slowly go away. But I think today it's a dead end because it does not contribute to the evolution of a virus because, as I said, the main selective force in viral evolution is to find a new host. If you go in the CNS, you can't get a new host. So these are all accidents. And viruses do not evolve to infect the CNS. They evolve to do other things so that they can find a new host. Next time, we're going to talk about the other type of infection pattern, the persistent infections where the viruses stay with you for your entire lifetime.